Okay, guys, in this second video for dihybrid crosses, we're going to do a sample problem. So we're going to look at a sheep, and we have two traits. We've got wool color and we've got eye color. So brown eyes and white wool are dominant, and black wool and blue eyes are recessive. So what we want to do is if we cross two heterozygous for both traits sheep, we want to look at what the phenotype ratio is going to be and what the total number of different genotypes is going to be. Now, usually for dihybrids, we don't necessarily look at genotypes, but for this first one, I think it's worthwhile. So in any genetics problem, you have to determine what your genotypes are and what letters we're going to use. So let's say for wool, white wool, we're going to make capital W white. And we'll make lowercase w black. And I'm going to make my my lowercase kind of round and my uppercase pointy because sometimes it can be difficult to tell these apart. And let's do capital B for brown eyes. And let's do lowercase b for blue eyes. So I guess I should say before we do our genotypes, we have to determine what our alleles are going to be. So if we have a white-wooled, brown-eyed heterozygous sheep, that's going to be capital, lowercase, capital, lowercase. And our cross is with another sheep that has the same genotype. So what we have to do is we have to remember, how do we find the gametes? So each parent has to give one trait. So the mother sheep can give a white allele and a brown allele, or she could do a white allele and a blue allele. She could do a black allele and a brown eye, or a black allele and a blue eye. So we have 4 different combinations that we see here. And again, we talked about this in the last video. So what I'm going to do is I'm going to write those 4 possible combinations along the top. For mom's gametes. And then we're going to do the same thing for our male sheep. And because it has the same genotype, we're going to have the same alleles in our gametes, or possibilities, I should say. And then down here, pull my camera down just a little bit. All right, in the interest of time, I'm going to pause the video and I'm going to pull all of my alleles down. So remember, just to refresh your memory, this comes down from the top, this comes over from the side. My capital comes down for the top, and my capital comes over from the side. All right, so you go ahead, pause the video as well, and finish filling in this Punnett square. Welcome back. Did you get your alleles filled in? So if you did, your Punnett square should look like this. So I want to start off by talking about the genotypes. And one of the interesting things when we do a dihybrid cross 2 traits is we start to see a little bit of a pattern. So how many different genotypes are we going to have? Well, we've got 1, 2, 3, 4, 5, 6, 7, 8, 9. So 9 boxes. There's a pretty good guess we're going to see 9. But there's a pattern to these. So you can see in these boxes in a diagonal, if I was playing bingo, this would be a diagonal bingo. I have the heterozygous for both traits. So over here for my genotype, I'm going to have heterozygous for both traits. And I'm going to have 4 of those. And then what I'm going to see is that as I come down the center, these are all going to be the individuals. So these are all the ones that are homozygous dominant and homozygous recessive. Now they're all different. So I have 4 different genotypes here. But what they have in common is that they do have a homozygous dominant, homozygous recessive, so on and so forth. So I can write these in. I have homozygous dominant for both, heterozygous or homozygous recessive for the first. Whoops, yeah, homozygous recessive for the second, and then homozygous recessive for both. And there's one of those. Well, that means my last two, I'm going to have two from each if I do the math. And what you're going to see is these two are going to be the same. So I'm heterozygous for the first trait, homozygous recessive for the second. I have two of those. And these are going to be the same. Homozygous recessive for the first, heterozygous for the second. I have two of those. And hopefully by now you kind of see a pattern emerging. Down here, these two are going to be the same. Whoops. Yeah. Okay, sorry, I thought I colored in the wrong ones. So here I'm homozygous dominant, heterozygous. I have two of those. And then for the last one, I'm just about out of highlighter, so I'm going to use this old kind of dried up marker. I have my last genotype. Heterozygous, homozygous dominant. Again, you're usually not asked to analyze genotypes for a dihybrid cross, but I thought for this first one, that would be a good thing to do. What you are asked to do very often is to analyze the phenotypes. So with 4 traits, my phenotypes are white, wool, and brown eyes. I could have white wool and blue eyes. I could have black wool and brown eyes, or I could have black wool and blue eyes. So those are my 4 possible phenotypes. So this is my wool color and these are my eyes. So let's go through and let's do hash marks to see what we're going to have. So over here, I have I have a white allele and a brown eye allele. So these 4, 1, 2, 3, 4, are going to be white and brown. Same here, I've got a white and a brown. Now here I have a black, I'm homozygous recessive, so this is going to be black wool with brown eyes. This is going to be white wool with blue eyes because I'm homozygous recessive for that. Here's my black and blue. Now I have white wool, blue eyes. I have black wool, brown eyes. Here I have a dominant for both. So I'm going to have 2 here and I'm going to have 2 here. And what we see is that when I add these up, I have 9 of the white brown. I have 3 of the white blue. I have 3 of the black brown, and I have 1 of the black blue. So we see this again and again and again. So for dihybrid crosses, for a dihybrid where both parents are heterozygous, the phenotype ratio, is always 9 to 3 to 3 to 1. Now, that doesn't mean that you can do that every dihybrid cross is like that. This is only when both parents are heterozygous. You'll always see this 9 to 3 to 3 to 1. One will be the double recessives. This will be dominant, and then in between you'll have one of the recessive traits with a dominant and the other recessive trait with a dominant. So what are our takeaways? Remember, these are mom's gametes. These are dad's gametes, and each parent gives 1 copy of each. So the reason we have a W and a B in each column is these are all the possible combinations that mom could give of her wool allele and her eye allele. And then the same thing for dad. We tallied up, we saw there's 9 different genotypes, really not worried about that too much, but here we have our phenotype ratio. And it's a good thing to remember that on a dihybrid where they're both heterozygous, you get a 9 to 3 to 3 to 1 ratio. Okay, that wraps up dihybrid crosses for us. Now it's time for some practice at home.